Hi everyone. For Chapter 11, we will discuss how gametes, which are your egg and sperm cells, are formed. They undergo a different type of cell division called meiosis. This lecture again will be critical to complete your lab exercise for Wednesday, so you have to pay close attention to all the differences mentioned in this lecture between mitosis and meiosis, because that's what's going to be asked on Wednesday's lab. You will be asked to also draw how chromosomes look like at different stages of mitosis versus meiosis. So, and that's what I will ask as well on the worksheet that will be due on Wednesday. So make sure you understand chapter or information from both chapter 10 and chapter 11. So if you didn't go through chapter 10 last week like I suggested, you will have to do it this week to catch up, okay? Because chapter 11 and 12 are two short chapters for a 2-hour, 40-minute lecture. 2 chapters will be covered this week. That's why I suggested for you to went over chapter 10 last week. So if you chose to squeeze 3 chapters today, that's not a scheduling thing, because chapter 10 should have been done last week already. Okay, so from chapter 10, we learned that mitosis is a type of cell division where daughter cells are exact duplication of their parent cell. But if all cells undergo mitosis, then technically speaking, we should be 100% clone of our parents. And yet this is not what we observe in the natural world. Even though we are born from cells that came from our mother and father, we are not duplications or clones of our parents. Even though we are talking about organisms of the same species, not every individual within the same species look the same. Humans, dogs, cats, primates, etc., they all look different from each other. Even though they're all dogs, we have different breeds of dogs, we have different breeds of cats, they have different colors, etc. This is what we have called genetic diversity. So, for this chapter, these 2 chapters on chapter 11 and chapter 12, we will answer these 3 questions. What contributes to genetic diversity? What gives us that genetic variations between different individuals within the same species? Why do we share some traits as our parents and some don't? And can we predict heritability? Can we put it into a math formula and calculate how likely or what probability you have of inheriting this particular gene from your parents? So these are the 3 questions we will be answering within this chapter 11 and chapter 12. In chapter 10, we talked about how gametes are supposed to be haploids, okay? Haploids meaning that they have half the amount of chromosomes. Remember, for humans, that is about 23 chromosomes. 23 chromosomes. Okay, 23 chromosome. And the 2 haploids, one from man, one from woman, sperm and egg, they will form a zygote, which is a diploid, which contain 46 chromosomes for humans, okay? For humans. And if chromosome number was not halved in our gametes, then we will end up with offspring that has more than 2 sets of chromosome, either 69 or 92 chromosome that we call aneuploidy, because you have either 3 sets or 4 sets of chromosome. And that usually, again, like I mentioned from last chapter, will not proceed with normal pregnancy, because a lot of chaotic thing goes on when you have DNA errors or chromosome errors like that. Now, remember that ploidy refers to how many sets of chromosome does a cell have. So remember, ploidy, sets of chromosome. Okay? All other cells that are not gametes are considered as somatic cells. which should have 2 sets of chromosomes, okay? 1 set from mother and 1 set from father. They are called homologous chromosome, as we have mentioned and discussed in chapter 10. Homologous chromosome means they carry the same genes but have different alleles or versions of the gene. Cell division of somatic cells involved 2 phases, as we have discussed in chapter 10, interphase and mitosis. Now, for cell division of gametes, which are your sex cells, egg and sperm, they also have interphase that is broken down into your G1, S, and G2 phase. Remember, that is your interphase of the cell cycle. Same as somatic cells, but they don't undergo mitosis. Gametes undergo a different type of cell division called meiosis, which is what allows diploid cells to become haploids, reducing the amount of chromosomes by half. Okay, remember, this is a very critical concept, that you're halving the amount of chromosome during meiosis. Now, for meiosis is able to reduce the amount of chromosomes by half because it involves 2 rounds of cell division, meiosis 1 and meiosis 2. Okay, meiosis 1 is the first cell division. Meiosis 2 is the second cell division. Notice that there is no another round of S phase for DNA duplication again. This is why we are able to reduce the amount of chromosomes in the second phase of meiosis. So from a diploid cell, you will end up with 4 haploid cells by the end of meiosis 2. For mitosis, however, remember that you start with 1 diploid cell and you end with 2 diploid cells that are genetically identical. Okay, so let me draw this for you so you know what I'm talking about. Okay, so for mitosis, you have 1 cell that started off with 46 chromosome, okay? After S phase, which is DNA synthesis phase, now you have 96 chromosomes because they're duplicated. And then you split them into 2 cells, which now both has 46 chromosome, okay? 2 daughter cells, okay, and they are genetically identical. Okay, for meiosis, on the other hand, and then what I would add here is mitosis is for cell division of somatic cells. Meiosis is cell division of sex cells. Okay, or gametes, another name for sex cells. Okay, so you still begin with 46 after S phase. You still end up with 96 chromosome, but you go through 1 round of cell division, which makes 2 daughter cells of 46 chromosome. This is meiosis 1. Here is mitosis. And for meiosis, you undergo another round of cell division that will give you a split of 23 chromosome. Again. So this is through meiosis 2. Okay? So notice that between meiosis 1 and meiosis 2, there was no S phase again. This is why you're able to halve the amount of chromosomes here. Okay? And what you end up with at the end of meiosis 2 is 4 daughter cells. And I will get to that point later in lecture, but they are genetically distinct. okay, or different from each other. So that's the main difference between mitosis and meiosis. Make sure you have your own version of this notes on paper as well as you're writing it. It also helps with committing to memory, okay? Just looking at pictures on the slide might not be clear enough, but when you actually draw this yourself, then you understand the difference between mitosis and meiosis, okay? All right, so just like mitosis and meiosis still has your 3 different phases during the interphase, which is G1, which is when the cell is growing, organelles are duplicating, they're getting ready to split into 2 cells. S phase, DNA replication is happening to duplicate the amount of chromosomes you have. And G2 is the final stage of interphase where you're finishing up and wrapping up all the organelle duplication, cytoplasm duplication, and also acquiring all the energy reserves to tell and allow the cell to split into 2. And just like mitosis, meiosis also have prophase, metaphase, anaphase, and telophase. But remember that they are all signified as part 1 because this is talking about meiosis part 1. Remember, meiosis has 2 rounds of cell division. So that's why each of the prophase, metaphase, anaphase, and telophase have a 1 after it. Okay, most of the events in these phases are the same between mitosis and meiosis. The difference between mitosis and meiosis 1 is that meiosis will create daughter cells that are genetically different from each other, meaning they will have different alleles or versions of the same genes. One sex cells may carry allele for blonde hair, for example, and another sex cell may carry allele for black hair because it is a mixture of the genes you have that came from your mom and your dad, okay? So that's why the daughter cells from meiosis are genetically distinct or different from each other. During prophase 1, you still have breakdown of the nuclear envelope and also chromosome condensation, just like mitosis, but an extra step called crossing over happens. For prophase 1 of meiosis 1, homologous chromosomes will line up together as a pair like this, and they will pair using a protein lattice structure called synaptonymal complex. This is what makes sure that the same genes are lined up against each other. If homologous chromosomes are misaligned, for example, here's one of your chromosome and somehow is not perfectly aligned and you have sort of that shifting in size, or even more extreme cases, if we're talking about something like this, okay, something like this, then this is what we call misalignment. It has to be perfectly aligned like this to make sure that gene on red chromosome is matching with the same gene on chromosome B, okay? So that way you don't end up mixing up wrong different genes with each other, okay? So you don't want different genes to swap DNA. You want the same gene to swap DNA during crossing over, okay? If the wrong genes was recombine or cross over, you will end up with missing or extra DNA segments on the chromosome. So this perfect alignment, as illustrated right here from this picture, is a very critical part of meiosis for production of gametes. This pairing itself is called synapsis. And within a synapsis, the synaptonymal complex will keep the homologous chromosome lined up against each other, matching perfectly by genes. And what happens between the chromosome is a process called crossing over, where segments of DNA are swapped with each other. They undergo a process of DNA exchange. Here's an example of what crossing over is. Okay, you have a paternal chromosome right here, and you have a maternal chromosome right here. Okay, so remember we talked about homologous chromosome is a pair of chromosome like that. Homologous chromosome. Okay, one of this, the red one, came from your mom, one of this came from your dad. They carry the same genes, but it can be different version of the same genes. So that's why here you have gene A, gene B, gene C, for example, and is represented by a big letter. Whereas on the maternal chromosome, you still have gene A, gene B, and gene C, but is now just a different version of it with represented by little letter. Okay, one example I've given is hair color, for example. Maybe this allele encodes for blonde hair color, whereas this allele encodes for black hair color. So now, when we're trying to form genes that will be passed on to the next generation, to your children, you will swap DNA between the two legs of homologous chromosomes so that you will have one chromosome that will have gene big A, big B, big C, okay? This chromosome now is different from this one, okay? Because it has swapped a section of DNA with the red chromosome. So now you end up with big A, big B, and little c gene for this particular chromosome. This particular chromosome, again, big A, big B, big C, okay? Now with this particular chromosome, when it becomes a gamete, they're going to have little a, little b, big C, because it has a little bit of blue chromosome from the blue chromosome, okay? Now this last one, chromosome right here, will then have little a, little b, little c gene. So you see how in the beginning they started off as just having big A, big B, and big C for blue chromosome. And then this one you started off with little a, little b, and little c. But because you swap or exchange DNA segment between the two chromosomes, you end up with a mixture where you have big A, big B, and a little c, or a big A, big little a, little b, and a big C. Okay, you will have to go through problems like this on the lab on Wednesday, so make sure you understand this part. Okay, all right, so hence, you have the resulting chromosomes after crossing over is each of them will carry some DNA from mom and some DNA from dad to pass on to their grandchildren. And this is why sometimes you don't see a trait in you that's passed from your mom, but you may see the trait from your mom in your children, because your DNA contains chromosomes from mom to your grandchildren, to her grandchildren or your children. Okay, so technically speaking, the resulting chromosome, okay, after crossing over, is each of them will carry some DNA from mom and dad to pass on to their grandchildren. And again, crossing over can occur anywhere along the length of synapse chromosome. It doesn't have to be between B and C. It can happen between A and B, okay? But it is what the same goal is that you will end up with mixing of genes, okay? And crossing over can actually also happens multiple times between the two homologous chromosomes during prophase 1 of meiosis 1. When we refer to a pair of homologous chromosomes lined up against each other, we refer to them as tetrads, okay? And after crossing over, alleles of the same genes are now mixed, and hence these chromosomes are no longer identical. Okay, they're not blue and red, but they have a little bit of mixture of genes from each other. This is one of the reasons why meiosis will result in genetically distinct daughter cells, not identical. This is why mitosis and meiosis is different, because meiosis has this extra step of being mixing the genes up. Okay, so let's go back to those concepts. Okay, I want to draw. Okay, here we go. All right, so here you have again your homologous chromosome. So when you have 4 chromosomes like that with each other, we call it tetrads. And then during prophase 1 of meiosis 1, they stack on each other like that, okay? The 2 chromosomes. And this stacking is called synapses or synapse, and is made possible by a protein lattice structure called synaptonemal complex. Okay, so make sure you understand what these terms means. Tetrad, synapse, synaptonemal complex. And then when the 2 legs, it's actually happening while it's being stacked like this, but the illustration always has it in this format to make it simpler and easier to see what's going on, is that you have this leg crossing over to the other side and this leg of the chromosome crossing over to the other side. So that's why you end up swapping the 2 DNA between these 2 legs of chromosomes. Okay, so this is called crossing over. And you're exchanging DNA content between the homologous chromosome. Okay. All right. So prometaphase 1 proceeds the same way as mitosis, where spindle fibers will still attach to each other, to each pair of chromosomes, using their kinetochores at the centromeres, as we have discussed in chapter 10. And during metaphase 1, what's going to be major difference between mitosis and meiosis is how the chromosomes are aligned at the metaphase plate. This is called random or independent assortment. Okay, here you have your, this diagram here is illustrating 4 chromosomes that are aligned one way. Okay, so one way of alignment. Okay, where you have these 2 sets of homologous chromosome right here and this tetrad of homologous chromosome right there. Well, another way they can align is this, okay? So this is another way of alignment. So one way of alignment is where you have blue chromosomes on one side of the cell and red chromosomes on the other side of the cell. But their positions can be swapped diagonally, where you have blue and red chromosome on one side of the cell and red and blue chromosome on the other side of the cell. So when they're aligned this way with blue on one side and red on one side, after 2 rounds of cell division from meiosis 2, you end up with daughter cells that has just blue chromosomes or daughter cells that has just red chromosomes. Because remember, whenever you split down the middle, okay, or whenever anaphase happens, you're always splitting the cells from the middle. So it's splitting the cell right there in that metaphase plate, and hence this is why you end up with just red chromosomes in one set of daughter cells and just blue chromosomes in the other set when they're aligned this way. Now, if they're kind of swap in order of alignment, then you're going to split again down the middle of the metaphase plate. And after meiosis 2, you will end up with daughter cells that has both blue and red chromosome and another set of daughter cell with different set of red and blue chromosome. So it depends on how they're aligned in the middle. You can end up with different daughter cells with different genes on there, different versions of the genes on there. Okay, so at the center of the cell, I already said how the homologous lined up can be rearranged into different combination. Okay, so that's what I went over with you in the diagram. Now, in reality, we don't just have 4, we have 46 chromosomes or 23 pairs of homologous chromosome. So there's going to be a lot more possible combinations to further mix up the genes. Okay, so let me draw this on, picture by picture so we understand what I'm talking about here. Okay, so in one case, this is metaphase 1 in meiosis 1. Okay, you have red chromosomes on one side and red chromosome on the other side. Okay, on the other side you have blue chromosome. Okay, blue chromosome, and then blue chromosome on one side. Okay, and when you split down the middle during anaphase 1, then you will end up with 2 cells. One cell has just red chromosome. Okay, the other one has just blue chromosome. Okay, you further split it in meiosis 2, and now you end up with 4 daughter cells that has just red chromosomes, single, because remember you reduced the chromosomes by half, and then you have just blue chromosomes on the other one. Okay, now the other way that they can be aligned during metaphase 1, okay, second case, is you have red chromosomes on one side, and then you have red chromosome on this side, and then you have blue chromosomes on this side and blue chromosome on this side, so that when you split the cell down the middle, okay, you're going to end up with daughter cells that has a mixture of your little red chromosome, and this one has the big red chromosome. The other one you have big blue chromosome and little blue chromosome. And during meiosis 2, you further split into your 4 daughter cells, and one set of daughter cells will have the little chromosome from this cell, and then you have the large blue chromosome. The other cell you have the little blue chromosome with a red large chromosome. Okay, so that's what we mean by independent assortment. Okay, independent assortment. This is what we mean, okay, when we're talking about independent assortment during metaphase 1 of meiosis 1. So independent assortment will determine which sex cell will inherit which alleles or trait from parent cell. And this is again reiterating that process here where you have, let's say, big red chromosome is for brown eyes and little red chromosome is for black hair. Okay, this big blue chromosome is for blue eyes and little blue chromosome here is for red hair. So this sex cells will become brown eyes, black hair offspring. This sex cell here will become blue eyes, red hair offspring. But if it's aligned this way, you're going to mix up that gene a little bit where now you have brown eyes, okay, chromosome mixed with red hair chromosome. And here you have blue eye chromosome mixed with black hair chromosome. So this individual is going to have a mixture of brown eyes and red hair, whereas this individual, if it becomes fertilized, will become a blue eye, black hair individual. So this is how independent assortment can mix up the genes a little bit from the paternal and maternal side, in addition to crossing over. So the possible number of genetically different gametes produced can be calculated using the formula 2 to the n power, where N is the number of chromosomes in a haploid set. So that's what N is, the number of chromosome in a haploid set. So for humans, our haploid number is 23. So the amount of different genetically different sex cells we can technically make is 2 to the 23rd power, which is equivalent to over 8 million possible genetically distinct gametes just from random assortment alone. We're not even taking into account the mixture, swapping of and exchanging of DNA segments during crossing over. Just from independent assortment alone, we can already generate 8 million different, genetically different sex cells, okay? Whether it be sperm or egg. So if we take into account of both crossing over and random alignment, during that, remember, crossing over happens during prophase 1 of meiosis 1, which allows DNA to be exchanged, and independent assortment happens during metaphase 1 of meiosis 1, where chromosome alleles will be mixed in terms of their position. It is almost impossible for two haploid sex cells generated in a person to be 100% genetically identical. This is what contributes to genetic diversity, which explains why when your parents gave birth to you and your sibling, if you have any, you and your sibling does not look exactly the same. You're not 100% clones of each other. One may have curly hair, the other may have straight hair. Or if you think about the case of fraternal twins versus identical twins, and you know what I'm talking about. Fraternal twins tend to look very different from each other because they came from two different sets of gametes, two different eggs that is fertilized by two different sperm. Identical twins are actually come from splitting of the embryo itself, not two different eggs, not two different sperms, okay? So that is why when you have two different eggs and two different sperms, they look very different from each other due to these two processes in meiosis in creation of your egg and sperm cells. The rest of meiosis 1 goes through the same events as mitosis, where you have anaphase 1, where the microtubules will pull apart the chromosomes from the metaphase plate. Telophase 1 and cytokinesis, the same thing. You have your cleavage whorl formation with your actin filament. Your separated chromosomes now arrive at opposite ends of the cell. Nuclear envelope starts to reform, and cytoplasmic component will finally pinch off from each other to create two new daughter cells with their own individual chromosomes inside, okay? So two cells with 46 chromosomes will result from the first stage of meiosis. So this is just from meiosis 1, okay? 46 chromosome, okay? And don't forget what's different between meiosis 1 and mitosis is the addition of two very important genetic variation steps, which is crossing over and independent assortment. Now, during meiosis 2, the two daughter cells undergo the process of cell division again, where the sister chromatids will separate and 4 haploid daughter cells will result by the end of meiosis 2. So here you have prophase 2, metaphase 2, anaphase 2, and telophase 2, followed by cytokinesis, and this is why you end up with 4 haploid daughter cells after meiosis 2. Now, for some species, there will be a brief interphase called interkinesis. This is different from interphase. It's called interkinesis just to mark the end of meiosis 1 and beginning of meiosis 2. One major thing to remember here is that there is no S phase, which means there is no DNA synthesis between meiosis 1 and meiosis 2. Chromosomes are not duplicated, and this is why when we split the cells a second time, we end up with cells that are haploids or contain half the amount of chromosomes, okay? as I have already drawn that out for you in the previous slide when I was drawing the difference between mitosis and meiosis. So the events that follows in meiosis 2, okay, you have prophase 2, metaphase 2, anaphase 2, and telophase 2 is exactly the same as mitosis. If a question asks for when in meiosis is exactly the same as mitosis, then that will be meiosis 2, okay? During meiosis 2, all phases, prophase, metaphase 2, anaphase 2, telophase 2, is exactly the same events that happens during mitosis. No further mixing of genes is necessary at this point because we already mixed it in meiosis 1, okay? So here's a good chart for you to keep in mind when studying for the next exam or working on the lab this week. Remember that you have mitosis from chapter 10 and meiosis from this chapter 11. In mitosis, you only have one cell division. You create two daughter cells in the end that are genetically identical. Both of them are diploids, and for humans, that means that they have 46 chromosomes. No genetic modification is made during mitosis. They are genetically identical, 100% clones of each other. For meiosis, you have two stages of cell division. 4 daughter cells are generated because you have 2 rounds of cell division. Each of those daughter cells are genetically unique due to crossing over that happens during prophase 1 and random assortment that happens during metaphase 1. At the end, each of those 4 daughter cells are haploids or contain just 23 chromosomes for humans. Okay, so this is a very good chart and table to remember for answering questions on the lab or for preparing for exam. So now I have a worksheet due for you that sort of lists the difference between mitosis and meiosis, and you have to draw what happens to the chromosome. This image here will be helpful right here. This is what I'm looking for on your worksheet. I will review the 2 processes with you here. Be sure to draw along at the same time with me so you understand and you're able to understand the difference between the two and are able to draw the same for your lab activity coming up on Wednesday and also the worksheet that is due. Okay, so make sure you grab a piece of paper, pause the video, ready, and it will be helpful if you want to use 2 colors to designate the difference of the homologous chromosome lining up side by side in meiosis. Okay, so before I wrap up with practice wrap up the chapter, I will draw that with you step by step. Okay, so for mitosis, okay, we'll begin with mitosis first. Mitosis. Okay, you have a cell, okay, and you have the beginning, one red chromosome, another red chromosome, okay, and then a blue chromosome, okay, like that. Okay, during interphase, the DNA content would double, and that's why by the time you are at prophase, okay, so this is interphase. After G1, S, and G2, you get to prophase. And then you would have your duplicated chromosome that are red and your duplicated chromosome that are blue. Okay? Now, in metaphase is when they would line up with each other, okay, in the middle towards the metaphase plate. Now, this is the difference in mitosis. The 4 chromosomes will not line up by homologous chromosome. They will line up all in a straight line, okay? There's no pairing of homologous chromosomes during mitosis, okay? They all line up straight just like that, okay? Line up in the middle. And then you would have mitotic spindle that's going to attach at the kinetochore from your centriose. And then during anaphase is when they will pull apart the sister chromatids. During anaphase, sister chromatids pulls apart. Okay, so you end up with pulling apart of the sister chromatid. Okay. Okay, and then you end up with telophase, which then you have your two cells about to be pinched off, one containing the original 2 red chromosome, and then the other, oh no, and then the other, okay, I'm just going to finish that off because you have that on your, as you're drawing with me. The 2 cells that's going to pinched off is you have your 2 red chromosome and then your 2 blue chromosome, just like in the beginning. Okay, so at the end of that was the end of mitosis. Okay, after telophase and cytokinesis. Okay, so remember the main point there is in mitosis. I'm just going to write that down really quick so you remember the major difference. In metaphase, you have all chromosomes line up in the middle. Okay, and anaphase is your splitting sister chromatids. Splits. Okay, so that was for mitosis. Okay, for meiosis, you have the you begin with the same cell with 2 red chromosome and 2 blue chromosome. Okay, and during S phase, after interphase, G1, S, and G2, you have duplication of the chromosomes. So you have 2 red and then you have 2 blue. Okay, during prophase. Okay, prophase 1. Okay, prophase 1. And then you have metaphase 1. So metaphase 1 is when they will line up in the middle. Now notice that they're not lined up in one straight line. They are now lined up according to homologous chromosomes. So how they can line up varies, as we have talked about. We're just going to go with one way where we have all red chromosomes on one side and all blue chromosomes on the other side. Okay, and they will again be attached at those mitotic spindles with the centrioles like that. So notice that this is already different. Okay, so for metaphase 1, they're lining up by homologous chromosomes. Okay, they're lining up by homologous chromosomes. And during anaphase, you're going to separate the homologous chromosome. You're not separating sister chromatids. You're going to separate the homologous chromosomes. Okay, so anaphase 1 separates homologous chromosomes. Okay, and then of course telophase, then you're going to end up splitting the 2 cells, which now has just red chromosome or just blue chromosome, because they have split it down the middle right here. Okay, so now it goes on to move on to meiosis 2. Okay, meiosis 2 is the same thing again. It doesn't have to go under interphase again. There's no S phase, no DNA duplication. So technically speaking, if this was prophase 2, then metaphase 2 is where now they would not line up by homologous chromosome, because they don't have homologous chromosomes now. They would just line them in the middle like this. Okay, and then you have your spindle fibers attached to the kinetochores of the 2 sister chromatids. And then it's going to move on to anaphase 2, which then will create not $2 cells yet. you have elongated cells because they're about to be split. Okay, you have red chromosome splitting on one side, and then the other one you have blue chromosome splitting. Okay, blue chromosome splitting. So again, this is splitting of sister chromatids, just like your mitosis anaphase. Okay, and telophase and cytokinesis will create 4 daughter cells, one with only red chromosome, half the amount of chromosomes, and the other one have blue chromosomes. Okay, and you end up with half the amount, which is 23 chromosomes at the end. Okay, 23 chromosomes. Notice that if you compared this set of daughter cells with this initial parental cell, you have 4 lines representing 4 chromosomes. Now you end up with 2, which is why meiosis 2, you end up with half the amount of chromosome. Okay, so you can now draw this whole process. I already draw the cell circles for you on the worksheet, and as well as the lab, they already drew in the circles for you. Like I said, those lab are not written by me, they're written by the department. So make sure you do the same thing on those circles, and that's why I created that worksheet, so you know what to do on the lab. Okay, all right, so that's what you're supposed to draw in terms of the difference between mitosis and meiosis. Now, if the lab asks you to draw the difference between 2 arrangement of independent assortment, then your other possible independent assortment right here is you have your metaphase 1. Your other possible independent assortment will be having red chromosomes on opposite sides and blue chromosome and on opposite sides. Okay, and that's how that metaphase 1 would look like if we're talking about different alignment due to independent assortment. Okay, independent assortment. And my handwriting is bad on the iPad, but you know what I'm talking about as I'm speaking those different words. Okay, all right, so make sure you understand that. I will post the animation video to eCampus as well, be sure to watch it so you can see the process visually. If you choose to not take advantage of the resources I provide online, then there's nothing else I can do. So make sure you take advantage of those things I post and these things that I go over with you while I'm drawing. Okay, this wraps up chapter 11. It's not a long chapter because we have already talked a lot about what's going on in cell division from chapter 10. Okay.